In that case, if you recall, those heterozygous individuals, get another pen. Had the genes on separate chromosomes. And so one of the things that we went through was that when you operate meiosis for two genes that are on separate chromosomes, because of the randomness with how the tetrads line up, and incorporating homologous recombination, which we don't talk about, just because it confuses things here, what you end up with is that mixture of big R, big Y, big R, little r, big Y, and little r, little y. And that's because of the law of independent assortment, that these genes that are on separate chromosomes assort independently of each other. But if you consider a situation where instead of the genes being housed on different chromosomes, what if instead those genes were on the same chromosome? So let's think about that. And I hope that isn't confusing. Genes can be on different chromosomes or they can be on the same chromosome. There's 20,000 genes, 25,000 genes in the human genome, and there's 23 different chromosomes. So the odds when you select any two genes are that they're probably going to be on different chromosomes, but there's a reasonable chance that you might choose two that happen to be on the same chromosome. So if you think about that situation, let's say this is a situation where we have those genes on the same chromosome. Now think about the pattern of inheritance that would result from this.
We're all thinking about it pretty hard.
You guys up?
Yeah.
That's great. No one is looking at me with confusion. They're looking at the... So what would you do to figure that out?
Draw meiosis.
You probably would draw meiosis, right? And so if you drew meiosis, you would probably draw something like this. I'll make my sister chromatid, and I'll make my other sister chromatid. And let's just say, hypothetically, that on this chromosome, there's one recombination event that occurs. All right? And that's not unreasonable. You might have one, you might have three. But if there's just one recombination event, the odds are good that for that one recombination event, it doesn't happen right in this region. It happens somewhere else on the chromosomes. You guys kind of see the logic behind that statement? If I randomly toss a recombination event onto these chromosomes, the odds that I toss that recombination event right there is very, very slim. And it's more likely it's going to occur somewhere outside of that region. So if you think about it, if you recombine at that point, what's that going to do to the genotypes of gametes that will be produced? What will the genotypes of gametes be?
Would it be in favor of the dominant gene?
It won't be in favor of the dominant gene. I mean, the genotypes, perhaps, yes, but I mean of the offspring. But think about the types of gametes that will be made. So go ahead and just take a moment. This is the question that's at the bottom of the third page of that activity we've been working through, where it says, but what happens if the two genes are right next to each other on the same chromosome? Practice sketching what will happen during meiosis.